name is Sarah Sprecher, and today I'll be presenting this paper I've selected from Nature Microbiology titled Wildfire-Dependent Changes in Soil Microbiome Diversity and Function. And I selected this article because I have a pretty solid background in soil microbiome analyses. I work in agricultural microbiology at my job, and so I am used to working with soil microbiome in typically normal and ideal circumstances, but I wanted to see how an extreme environment such as a wildfire could impact the functionality and diversity of a soil microbiome. And if you're someone who's interested in the soil microbiome or in the microbiome of extreme environments, then this will be right up your alley. A bit of some backstory for my selection of this article, and also as a personal anecdote, my connection to this topic is pretty deep. I'm from a state that endures fire season almost every year, and some years are more severe than others. But 2020 was one such year when we had our most devastating fire season on record. And the Santiam fire photo pictured here is actually just one of several very large high-impact fires that were in the state of Oregon. And they all collectively burned 11% of the Oregon Cascades and forced about 40,000 people to evacuate. So fires in Washington, Oregon, and California are all very extensive, very frequent, and contribute to a lot of damage of ecosystems and personal property. One last thing to note too is that the air quality index from this fire that happened in 2020, from all the fires, caused the worst ever recorded air quality in several big cities, including Portland. So for those of us who were not impacted by evacuations, there was still a concern for air quality, which I think is very significant as well. Urgency of this topic, I pulled another research article that was examining the intensity and frequency of wildfires over the years as a result of climate change. And here they show that 21 years of radiative power, or fire radiative power, or FRP, released by wildfires can be observed from satellites and then overlaid to highlight increased frequency around the world. They only used wildfires that were exceeding the 99.99th percentile of very severe high radiative power, and in that it shows which fires were most notable, most damaging, and between the years of 2002 and 2023, they report a 2.2-fold increase in these high-intensity, very high-severity wildfires. And I think it's useful to note here that it's not just a problem in the United States. We also see this in other countries as well around the world. So definitely not something that is only affecting one place in the world. As we face more wildfires due to climate change, I think it becomes necessary to have a broader understanding of the forces at work as a result of a wildfire in an ecosystem. And the authors had this same motivation to do this study, and they use a genome-resolved multi-omic approach in order to effectively tackle the issue from all angles. So the main question I think they try to answer is, what effects do wildfires of any severity have on functionality and microbial diversity of a soil microbiome? And I say that it's in mostly high elevation coniferous forests because that's where they got their sample set from, but they note in this paper that there were two other past studies that applied genome-resolved metagenomic analyses as well. One was to a Mediterranean basin in Spain, and the other was a boreal forest taiga in Alaska. And I think that this approach they wanted to use to just further characterize the soil microbiome. And the 2 previous articles that did use these approaches came out in 2014 and 2015. So by publishing this paper later on, they may be able to use some newer technologies and kind of get a better idea of how all of these different parts and different players like affect the full image. So the bottom line up front of this paper is that wildfires alter and shape the functional state of the soil microbiome. And additionally, I think it's important to mention that they saw increased burn severity lead to loss of diversity, as well as less complexity in community structure. And although those may sound not too surprising, we're talking about something catastrophic happening to a normal soil microbiome. I do think it's important to note that there is a lot more context that can be provided in these situations. And I think that the authors do a pretty good job of trying to figure out what can enrich this understanding that we already have, which is that, we lose a lot of microbial components of a community when something as catastrophic as wildfire happens. But what does that actually have to mean for the microbial processes that continue? And in some other points, I noted that they compared surface soils with deep soils, and they saw a muted response to wildfire in deep soils, which they proposed could be because of the sort of the buffering effect that the depth of soil would have. They also did a bit of viromics to analyze how viruses are affected by wildfire, and they saw similar trends in viruses to the bacterial and fungal communities, and saw a homogenizing influence from wildfire, that is, in soils that had more severity wildfire, they had more of a homogeneous community structure versus like in deep soils and less severity wildfire soils. And additionally, they were able to identify certain pyrophilus phyla that were very abundant in soils that have faced wildfire. And interestingly, I saw that the previous 2 studies that used the multi-omics approach that they did to analyze the soil microbiome also recovered the same taxa that they illustrate in this article from burned soils. So I thought I think that the fact that they're in agreement is actually very interesting. And lastly, the genes that these bacteria are encoding in the post-fire soils were shown to be very important for heat tolerance and fast growth of the bacteria. So this could illustrate some different traits that maybe are selected for in these post-fire environments where the bacteria that have the possibility of encoding these genes may be able to outlive the taxa that are not able to survive in these conditions. So I thought that was very interesting to look into as well. So for their sampling methodology, they collected their samples from 2 fire sites that were along the border, called the Ryan Fire and the Badger Creek Fire, which both happened in 2018. So they collected these in 2019, 1 year on. And they sampled from 4 replicate burn severity gradients, and 2 of these were in the Ryan Fire site, and 2 of these were in the Badger Creek Fire site. And the burn severity gradient looked like, as you see here, a control unburned soil, and then low, moderate, and high severity soil burn. And from each burn condition, they took 6 subsamples, so they ended up with a lot of soil samples. They had 176 total collected, which I think is very useful to really highlight what's going on in all these different wildfire sites. And I said before that they used a multi-omics approach in order to really have a deeper understanding of everything that's going on. And they mainly used marker gene analyses to profile bacterial and fungal communities, which they did via 16S rRNA sequencing using the aluminum HiSeq paired end reads, and then using some other additional software. They used QIIME2 to process the reads, and Dada2, which is a de novo approach for denoising, and coupled with the databases Silva and Unite so that they could assign taxonomy, and then some packages for analysis. rVegan is actually very important for ecological analysis, and they also used PhyloSeq, and this allowed them to conduct non-metric multidimensional scaling on Bray-Curtis dissimilarities in the bacterial groups that they discover, and then they were able to do some metagenomic analysis. They created these MAGs, which are metagenome assembled genomes. They used a subset of 12 of the Ryan fire soil samples, which came from places that had like the highest severity of wildfire, and the data that they used from these MAGs, they could assign taxonomy and that way see who's present and what they're doing, and that kind of became the bulk of this article's work. And the metagenome data that they compiled actually exists on NCBI under this bioproject code, so that can be looked at, and you can download the files and do your own analyses if you'd like. And this was collected by Colorado State University, so that is also why it says that there. For the results, there were a lot of figures in this article, and I won't have time to get through all of them, but I tried to focus on the ones that are most fundamental. And this one here highlights the percent change in relative abundance from unburned control samples to soils that had some severity of burning, so low, moderate, or high. And the asterisks on the bars denote significant differences. So most of them, there was a significant difference. And you can see the phyla on the x-axis. Most of them are under 0, so they actually had a loss of diversity because of any severity of fire. And the ones that are above the x-axis bar, those actually rose their relative abundance. And the interesting thing here is that the firmicutes showed the largest increase, but the authors do note that the firmicutes still did not have an overall high relative abundance in the soil microbial community. They were still pretty low. But the key players that we'll talk about that were most abundant, those can be seen more on the left side of the graph. And we'll get into those and why that they're somewhat the important part of this paper and understanding what they're doing. Here they used non-metric multidimensional scaling for their bacterial and fungal communities at the surface soil depth and the deep soil depth, which is denoted by shape, and then the burn severity which is the control or unburned, and low, moderate, and high burn severity is denoted by color. And I think the main thing that this illustrates is that the increase of burn severity leads to more similar community structure, which you can see in the clustering of the orange and red shapes. But this is a lot more impactful in the surface soil depths, which you can see with the triangles, and with the deep soil depths, which is the circles, there really wasn't a lot of influence or as much as the surface. So they kind of highlight that this could be maybe a dampening effect in the deep soils, potentially due to insulation, whereas the surface soils would receive the most impact from the wildfire. And another thing to note too is that the fungal communities did mirror the bacterial communities, and they saw that there was some diversity loss associated in both due to any increase of burn severity. This is another view of the surface and deep soils, which I think shows that there is an increased separation of burned and unburned communities, but it's much more prevalent in the surface soils than it is in the deep soils. And they see this also reflected in the fungal communities as well, but overall they highlighted that the deep soils showed a greater community dissimilarity. So there really wasn't much homogenizing there from the wildfire at any level of severity. That they performed a viromics assessment of the soil samples, and it was interesting to note that the same trend that was seen in the bacterial and fungal community structures was also seen in the viral communities. Both DNA and RNA were less homogeneous in deep soils compared to surface soils. So the darker red and the brown color would be the deeper soils, and there's not really much of a grouping for those versus the red and yellow, which would be the surface soils, are a little bit more clustered together. And so when considering who is present in these burned soil environments, I think it's very useful that they did a phyla distribution of their collection of mags, which I talked about previously, the metagenome assembled genomes from their subset of 12 Ryan fire samples. And they this produced 637 total mags, which I have highlighted in the top right. And looking at the distribution, you can see that Actinobacteriota dominated the post-fire soil microbiome in this study. And it's worth noting that this finding is also reflected in the other two studies that I talked about previously that also used similar approaches. They saw very much Acetobacteria and Proteobacteria in their wildfire soils. And so the Actinobacteria became most abundant 1 year on. But if you remember from the previous slide, or from the one of the previous speakers, that the firmicutes had the greatest increase in their relative abundance. But as you can see now, the firmicutes really don't represent that much of the full picture. So I think that's kind of an interesting question, though, because they did see the largest increase in their relative abundance, and the fact that they did, even though they're such a smaller represented species in the overall picture, that could be something worth looking into, because that percentage is way higher than all the others that were demonstrated in this study. So I just thought that was something interesting to note. So they wanted to look at the gene expression of these post-fire mags, the ones that were showing the best survivability. They were most abundant in these soils. They wanted to look at gene expression to see if there's a significant increase in a kind of gene that allows them to have better survivability. And so they actually took a pool of 40 of the mags that were most abundant in high and low severity soils. And spoiler alert, 28 of them were actinobacteria, which we saw was the most prevalent phylum. And they saw that 38 out of 40 of these mags encoded sporulation genes, which if most of these are a spore former, I think that could really highlight the importance of being able to form spores to survive very damaging wildfire conditions. And they also saw that 16 out of 40 of the mags had genes for mycothial biosynthesis, which they said could be used for thermal resistance. And what I wanted to note with these figures here is that they saw 22 of the mags had faster doubling times. And they looked at the 4 different conditions of high severity surface soils, high severity deep soils, low severity surface soils, and low severity deep soils to compare the doubling times of these mags. And the only one that had a significant trend was the high severity surface soils. So the authors allude that these conditions specifically might select for microorganisms that can grow quickly. Because if you think about it, being able to outgrow and outcompete essentially the other taxa that are there could definitely give you an edge to surviving wildfire and the aftermath of a wildfire. They also saw that mags recovered from the high severity surface soils had significantly higher guanine and cytosine content, and having higher GNC content has actually been linked to thermostability from other articles. So I think could be something interesting for why these taxa are specifically able to survive. And you can see in the box here that's red for high surface soils that it had the greatest mag GC content out of all of the conditions, even more so than high severity deep soil or low severity surface soil. This is the same slide showing the bottom line up front as I did before, but I really thought that these figures did a good job of showing the how the functional state of the soil microbiome is altered and what could be happening at different levels of the microbiome and what could be happening in the various communities like bacterial and fungal and viral, and how they can kind of relate to the recovery of the soil microbiome environment. And I wanted to just highlight it here, and I think that the authors did a very good job of showing this kind of all-inclusive understanding of what all these different players are doing. For why is this important? I think we've seen that wildfires reduce the microbial biomass of the soil microbiome, as well as the diversity of the bacterial and fungal communities. It also creates an environment where survivability through certain genes and traits is favored, as we've seen. And I think that this can be very important to providing some insights into post-fire plant recovery, seeing as how most of the changes that were observed would directly influence that. So I think that knowing more about how these microbial processes are disrupted could be very key to understanding more about the effect of wildfires at the micro scale. So I think the main importance of this paper is that how the soil microbiome is functioning drives how the forest ecosystem is functioning. And a disruption like wildfire fundamentally changes how interspecies interaction occurs and the biogeochemistry of the environment that they inhabit, and also the activity of the soil microbiome after fire is very important to the regeneration of the ecosystem. So I think it would logically make sense that to have better understanding of how an ecosystem is able to revegetate after a wildfire is based in how the soil microbiome is able to overcome such an event. So some questions I just wanted to leave the club with is, would we expect to see these results in other forest microbiomes? And I think that this could be interesting because, as I said, there were 2 studies that also looked at wildfire impacted soils with a multi-omics approach, but it was in the Mediterranean basin, and then the other one was in the Alaskan taiga forest, and this one was obviously in high elevation coniferous forest. So I think it could be interesting to see if the same results are replicated elsewhere. And then my next question is, could pyrophilous taxa, such as actinobacteria, could those be used to help an ecosystem recover faster? I think maybe if there was some way to form a bacterial inoculant, that could be something that if we found that these were actually very helpful bacteria for the community, it'd be interesting to see what kind of work they could lead themselves to. And I had also talked about how deeper soils had sort of a muted response to the wildfire. And I was kind of curious if they would see this trend continue with even deeper soils. So the deep soils they looked at were between 5 and 10 centimeters, and so I wondered if maybe if it was deeper than 10 centimeters, if they would see even less of a response to wildfire. And the fourth question would be that I had was, does the diversity loss have a huge effect on the soil microbial processes? And I think the short answer would be yes, but then the long answer is, but how? And I think that would be maybe the next step to understanding more about how the biogeochemical processes are also interrupted. And lastly, what are some other genes that these microorganisms are encoding that could help them thrive in post-fire soils? And I think that some of the ones that they brought up are very interesting. I don't think it's the full picture, and I think that there are other genes that we could also look at as well. These are just some of the references I used in my presentation. Thank you so much for listening to this, and I hope that you found this as interesting as I did.